Today, topic of our discussion is biomolecules. What are biomolecules? Well, biomolecules are derivatives of simple compounds of the nonmetals, and that are oxygen, hydrogen, nitrogen, sulfur, and phosphorus, for example. When hydrogen is added to oxygen, water will be formed, and when hydrogen combines with nitrogen, ammonia will be formed. Let's balance these equations. Before moving into detailed discussion of structures of biomolecules, let's clear our basic concepts. Always remember that carbon has 4 valencies, nitrogen has 3 or 5 valencies, hydrogen has 1 valency, and oxygen has 2 valencies. Are you familiar with the term alkyl? Well, this is the structure of alkane or methane. Removing 1 hydrogen makes it methyl. It's an alkyl group. Now, what is an Aryl group? Well, from aromatic ring, removing 1 hydrogen makes it Aryl group, and they can be named according to types of group attached on them. For example, 1 hydrogen removed from aromatic ring and replaced by CH3 group makes it toluene, and chlorobenzene will be formed if hydrogen of aromatic ring replaced by chlorine. In carbonyl group, carbon atom is double bonded to oxygen atom. So, keep this carbonyl group in mind. Aldehyde will be formed when to carbonyl group if hydrogen and R group are attached, and ketamine will be formed in structure of aldehyde if hydrogen is replaced by R group. Carboxylic acid will be formed if in the structure of ketone, 1 R group is replaced by OH group. In primary amine, 1 nitrogen, 1 aryl group or alkyl group, and 2 hydrogen groups are present. In secondary amine, 2 R groups and 1 hydrogen is present, while in tertiary amine, 3 hydrogens are replaced by 3 R groups. In structure of primary amide, 1 R group is present, and 2 hydrogens are attached with nitrogen, but the presence of carbonyl group makes it different from amines. In structure of secondary amide, as compared to primary amide, 1 more hydrogen is replaced by R group. So total 2 R groups are present. In structure of tertiary amide, 1 more hydrogen is replaced by R group as compared to secondary amide. So total 3 R groups are present. Let's discuss the structure of primary alcohol. All in alcohol indicates OH group attached to carbon. This carbon is attached with further 2 hydrogen atoms, and R group and 4 valencies of carbon are fulfilled. In case of secondary alcohol, 1 hydrogen of primary alcohol is replaced by another R group, so total 2 R groups are present. In case of tertiary alcohol, 1 more hydrogen from the structure of secondary alcohol is again removed and replaced by R group to form tertiary alcohol. Ethyl is class of compounds that have 1 oxygen atom bonded to 2 alkyl or aryl groups. There are 2 types of ether. In symmetrical ether, R groups on both sides of oxygen are same, but in unsymmetrical ether, R groups on both sides of oxygen are different. In the structure of ester, it has carbonyl group attached with R group on one side and OR group on another side. R group can be alkyl or aryl. Now moving towards carbohydrates. They are also known as saccharides. Firstly, let's discuss monosaccharides. This is the structure of glucose. It has 6-membered ring. Let's give numbering to carbons 1, 2, 3, 4, 5, and 6. But fructose has 5-membered ring. Let's start its numbering 1, 2, 3, 4, 5, and 6. This is the structure of galactose. Difference of glucose and galactose is in the arrangement of OH group at position 4. These monosaccharides combine to form disaccharides. For example, glucose combines with fructose by removing 1 hydrogen from fructose and 1 OH group from glucose to form sucrose, and water molecule is released. So sucrose is formed. Another disaccharide is formed when galactose combines with glucose through the removal of hydrogen from galactose and OH group from glucose with release of water molecule, and lactose is formed. Next in carbohydrates are polysaccharides, and its example includes starch, in which many glucose molecules are attached with each other. Other examples of polysaccharides are glycogen and cellulose. Another type of biomolecules are proteins. They are made up of amino acids. The basic structure of amino acid consists of amino group, side chain, and carboxyl group attached to it, and a hydrogen to fill the valency of carbon. How does a peptide bond form? Well, consider two amino acids, OH group from carboxyl group of one amino acid removes, and hydrogen from amino group of another amino acid removes. A peptide bond will be formed, and a water molecule will be released. For detailed video on structures of lipids and cholesterol, watch our video on phospholipids and sphingolipids, and video of cholesterol. Now let's understand the structure of nucleic acids. They are made up of nucleotides. Do you know what is the structure of nucleotide? Well, it has a pentose sugar. That means 5 carbon sugar. 1, 2, 3, 4, and 5. A phosphate group, and a nitrogenous base. There are 2 types of nucleic acids, deoxyribonucleic acid and ribonucleic acid. Let's understand the structure of DNA. Nucleotide, well, it has nitrogenous base, pentose sugar, and a phosphate group. RNA. Nucleotide has a nitrogenous base, phosphate group, and a pentose sugar. What's the difference in the structure of DNA? Nucleotide and RNA. Nucleotide, so the oxygen of OH group at position 2 is removed. That's why it is called as deoxyribose. While in RNA, nucleotide, OH group is present at position 2. That's why it is called as ribose. Let's understand the structures of nitrogenous bases. It is adenine. It has 2 rings. One is 6-membered, and other is 5-membered. Let's start its numbering. 1, 2, 3, 4, 5, 6, 7, 8, and 9. As name indicates, nitrogen is present in its structure, as it is a nitrogenous base and a amine group at position 6 and R group at position 9, double bonds between position 8 and 7, 2 and 3, 1 and 6, 5 and 4. As compared to adenine, in structure of guanine, amine group at position 6 is replaced by carbonyl group. Amine group at position 2 is also present, but double bonds are between position 7 and 8, 2 and 3, 5 and 4. But no double bond between position 1 and 6. This is structure of cytosine. It has 6-membered ring structure. Let's start its numbering. 1, 2, 3, 4, 5, and 6, double bond at position 5 and 6, 4, and 3, a amine group at position 4, and carbonyl group at position 2, but in uracil, amine group at position 4 is replaced by carbonyl group. So total 2 carbonyl groups are present, double bond at position 6 and 5, but not between 4 and 3. So a hydrogen is attached to nitrogen to fill its valency. This is the structure of thymine, double bond between position 5 and 6. It also has 2 carbonyl groups, hydrogen attached to nitrogen of position 3, but a methyl group is also present at position 5. That was all about today's video on biomolecules and a detailed discussion of their structures. Stay tuned to sqadia.com for more precise and knowledge stuff videos.